We really don't know how cellular life first appeared on Earth, but one of the most accepted hypotheses is that the first protocells were formed by lipid vesicles containing self-replicating macromolecules that could transmit information. But replication of such biomolecules is problematic because we know that information is transmitted in the DNA via RNA to make proteins or other functional biomolecules. But that information has to be decoded by some very specific protein complexes. But proteins, of course, do not carry genetic information, so they cannot have been the first biomolecules in those protocells. And there is no evidence for DNA being able of self-replication. So this chicken and egg problem was partially solved when scientists discovered that RNA can perform both functions. It can carry genetic information in its primary sequence, the order of nucleotides, but also can behave as an enzyme carrying out some biochemical reactions that could explain its self-replication. Thomas Cech, working with a protozoan called tetrahyamine, and Sidney Altman, working in bacteria, identified catalytic RNAs, later called ribozymes, which are RNA molecules capable of catalyzing specific biochemical reactions, including in some cases their own synthesis. Group 1 introns, for instance, are found in bacteria, some lower eukaryotes and plants, and behave like ribozymes capable of self-splicing. These discoveries led to the hypothesis of an RNA world in which the first protocells would carry ribozymes. All the molecular fossils remind us of this RNA past in modern cells, such as the ribosome. The ribosome is a large RNA molecule required for the translation of RNA into protein. Although today ribosomal RNA needs to form a complex with proteins in order to carry out translation, it has been shown that the catalytic part of the ribosome is precisely the RNA component, with proteins playing mostly a structural role. So the ribosome can be considered as a ribozyme. A testimony to the RNA past of all cellular life on Earth is the fact that it is possible to establish the relationship between all life forms comparing the ribosomal RNA of various organisms, a breakthrough achieved in the late 1970s by Carl Woese. It is not clear how the transition from an RNA world to a DNA world could have taken place, but the tree obtained by Woese tells us about a looker, a last universal common ancestor of all cellular life forms, although we still know nothing about what this ancestor was like. This three-domain tree shows archaea and bacteria, which are very similar biologically, together with eukaryotes, with leuca at the root. Later phylogenetic studies comparing all the genes shared by all living beings found the same tree with three domains, but more sophisticated analyses placed the last universal common ancestor, the leuca, between two main branches, one leading to bacteria and the other to archaea plus eukaryotes. This is of course a simplified view, because there is evidence that things at that time were not so simple in terms of exchange of genetic information.